7 repeats, codons, coding glutamine. If the timing of onset was determined by the polyglutamine length estimated by the CEG sizes, these guys with double duplication for identical numbers, yeah, of the CEG sizes, they would drive earlier onset than the others because they have 4 more glutamines. But the data is showing exactly the opposite. So this turns our expectations upside down. And this finding necessarily has to change the explanatory hypothesis for the disease. Michael Hayden's group found the same among symptomatic HD of affected subjects from 6 families with low penetrance alleles, actually. All index cases carried loss of interrupting CAA, I mean. All of them carried this kind of alleles. And more importantly, the average age of onset of them was 25 years earlier than that predicted by the CEG expanded repeat. This study went further and found an association between carrying or not the interruption. Those guys carried the interruption, they have the canonical form of the allele, and those ones carried this allele without interruption. And they found this association with the peripheral mosaicism of the CEG expanded repeat. Those guys without interruption have larger mosaicism than the other subjects. So how do DNA repair defects fits into this hypothesis? So according to the Juwas from the GEM HD group and also from the Betancourt and co-workers, DNA repair genes are the strongest modifiers of the age of onset in Huntington's disease. And actually, they are important in modifiers of the velocity of the disease progression as well. This is very interesting. So how do DNA repair defects fit into further expansions? I will use the figures presented by the Huntsman-Moss paper to try to reply. So they say that the CEG repeat DNA, when it is partly unwound by transcription, might be recognized by a complex of proteins of the DNA repair, which recruits the endonucleases that cleaves DNA. And then the repair of the break leads to further expansions of the CEG repeat. As time passes, expansions occurs inside the neuron and might be related to the neurodegeneration and the starts of the symptoms. So therefore, relationship between DNA repair and somatic mosaicism in animal and cellular models put former evidence of mosaicism obtained in humans in a favorable light. So somatic mosaicism in humans has been described since the 90s. However, unfortunately, many researchers wonder if technical problems would not explain these findings at that time. For instance, in 2007, large somatic expansions of more than 100 repeats were seen in the affected gray matter of Huntington's disease carriers. 2 years later, Suomi and co-workers related somatic mosaicism to severity. They found that the brains of Huntington's disease subjects with early onset had much more further expansions of the expanded CEG repeat than the brains of the extreme late onset. In this pair of early and late onset brains, both have 47 CEG repeats. You can see that the early subject, the subject with the early onset, have much more changes in the repeat size of the inherited allele in gray than the late onset. So in 2019, 10 years later, the same reports that associated the pure tract that I already talked to you with the HD severity also studied somatic mosaicism. In the Ciosi and co-workers, simplifying a lot, I would say that the number of the somatic expanded alleles minus the number of inherited alleles as measured in leukocytes, of course, equaled what they called somatic expansion score. What they saw is that the carriers with an excess of expanded alleles in yellow and in blue, of course, had earlier ages at onset than the other subjects. Okay, but the evidence linking DNA repair and mosaicism in HD patients, is this already obtained? Well, I would say that there are quite a few associations studied so far, but the hypothesis of an interrelation between them is recent, so we need to wait a little bit more. So the same somatic expansion scores proposed by the group of Moncton was associated with SNPs in these genes related to the DNA repair, FAN1, for instance. Yeah, they presented us only the p-values, unfortunately. But the group of the JMHD consortium also estimated the expansion sizes, further expansion sizes after the inherited expanded one in blood. And they found that the MSH3 allele associated with the increased expression of this DNA repair protein, MSH3 protein, was more frequently found in HD subjects with the highest expansions scores in blood, those guys in blue circles in the cartoon. So all this puzzling data led researchers to interpret the results as occurring in the following proposed order of events. We are not sure of that, but this is the proposal. The CEG repeat before the interruption is exposed to repair defects throughout life. And this effect would be worse if the tract is pure, but this is rare, okay? So then repair defects would lead to a continuous somatic mosaicism towards greater expansions. In post-mitotic cells, it would be more important. Symptoms then would start when the neuronal CEG expansion crosses a threshold that would be greater than the estimated blood from the CEG repeat in peripheral blood. So you can agree with me that the sequence recapitulates almost exactly what Kaplan and co-workers presented as the universal mechanism of the disease related to repeat expansions. So this is the hypothesis, and so this is the breaking news, a new hypothesis. You can ask me if this hypothesis is already consolidated or definitive, and I would say not yet. But the fact is that this hypothesis better explains several phenomena. And of course, there is a number of questions that need to be answered. For instance, is somatic expansion required? And does size matter for neurodegeneration? How does the pure CEG trigger more damage through repair in spite of being rare? We don't know. By what mechanism would the expanded CEG repeat crossing the threshold do damage? By something related to RNA toxicity, RAN translation, only because the polyQ would be very large? And how to combine and balance the effects of potential nuclear toxicity with the certainty of toxicity due to polyQ? Well, I hope I've helped in the debate about what's new about genetics, but I need to leave these questions for you. And I conclude by stating that these recent findings acquire enormous importance because they open up new therapeutic approaches associated with the presence of the interruptions in the CEG tract. We will hear about this later today, okay? So, but I speak it here on behalf of my neurogenetics study group in Porto Alegre, south of Brazil. We live here, we work here. That's it. I thank you for your attention, and I will be available for questions.
Okay, hello. I'm going to start with this disclosure slide.